There are 7.8 billion people living on the planet, which means there are 7.8 billion different existing genomes. This large disparity between individual genomes brings up the question: how can the small number of medications available be compatible with the 7.8 billion genomes on this planet? The simple answer is pharmacogenomics. Pharmacogenomics is the broad study of how genes affect a person's response to drugs. Pharmacogenomics includes the study of how all genes in the human genome interact with all different types of medications. It aims to develop effective and safe medications in suitable doses by identifying a person's genetic makeup and cultivating medications based upon that makeup. Overall, the long-term goal of pharmacogenomics is to help doctors identify which medications are suitable for which type of genome.
Everyone's genome is different, even the genome of identical twins. This can be attributed to genetic variations. The most common type of genetic variation is a single nucleotide polymorphism, or an SNP, which can also be pronounced as a SNP. In order to understand SNPs, we need to take a quick look at what our DNA looks like. A strand of DNA consists of a phosphate group, the sugar deoxyribose, and the nitrogenous base. A SNP is a difference in a nitrogenous base. For example, in a strand of AATTCGA, a SNP would be where the nucleotide cytosine replaced the nucleotide adenine. This is also known as mutation. Mutations generally carry a negative connotation since they're associated with genetic disorders. However, SNPs usually don't cause any harm. In fact, there are around 4 to 5 million SNPs in a person's genome. They are also helpful as biological markers that allow scientists to identify genes associated with a disease.
Arthritis is a condition that affects 23% of adults in the United States, which is about 54 million people. It is mainly characterized by joint inflammation, which can lead to painful or stiff joints. Arthritis can present itself in several forms, the most common form being osteoarthritis. This is a disease which occurs when the tissues of the joints begin to break down, causing pain and stiffness, commonly in the fingers, knees, hips, neck, and lower back. The actual causes of arthritis are not well understood. Arthritis can be caused by poor lifestyle choices, which lead to obesity or being overweight, as this can cause inflammation in the joints. However, it can also be caused by a specific gene which has been passed down from parent to offspring. This gene is called the HLA-DRB1 gene, and it can be targeted to treat arthritis.
Current treatments for arthritis are immunosuppressants and tumor necrosis factor inhibitors, but these agents can be ineffective in up to 30% of rheumatoid arthritis cases. These treatments are found through a trial and error process because there's a lack of biomarkers or signs in the gene to discover an individualized treatment. Pharmacogenomics can analysis finds changes within specific genes such as HLA-DRB1, and pharmacogenomics treats patients based on these SNPs. One promising pharmacogenomic treatment using the aforementioned variables was found through research on rheumatoid arthritis synovial subtypes or joint subtypes. The subtypes can tell the scientists about. Currently, researchers are finding blood biomarkers that can correlate with the synovial phenotypes to predict outcomes to treatments. It's amazing how such a field as new as pharmacogenomics can be used to create a multitude of prospective treatments that target arthritis and many other conditions.